We are so excited about today's presentation titled The Need for Speed. We're going to learn about the burrowing behavior in the Pacific sand crab. And here to tell us all about these cool creatures, we have Dr. Gita Kolluru. She obtained her undergraduate degree in biology from Queen's University in Canada and a master's and PhD in biology from the University of California, Riverside, where she studied mating behavior in tropical fishes and crickets. She went on to be a postdoctoral researcher at UCLA and a lecturer at Humboldt State University. She is currently a professor in biological sciences at California Polytechnic State University, San Luis Obispo, where her student-centered research program focuses on mating and aggressive behavior in Cuban freshwater tropical fishes. She teaches courses across the curriculum in ecology, evolution, and behavior. You may also catch her on Animal Planet TV shows talking about puzzling animal behavior. We are so excited for today's presentation. Again, thank you so much for joining us.
Thank you so much. Thank you, Robin, for the introduction. And thanks to everyone who's here and who may watch this. I'm going to go ahead and share my screen and then turn off my video. So thank you all for coming. And as Robin said, I'm going to be talking about burrowing behavior and some other aspects of the biology of our local Pacific sand crabs. And they're a really fun animal to work with. Probably many of you have played with these crabs at the beach, and so they're familiar to a lot of us who live here, especially people who've grown up in this area along the Pacific coast. So I do want to start by saying that the beach can be a very dangerous place. And I'm not talking about the things that you can see in this image. I'm talking about what's going on down here in what we call the swash zone of a sandy beach. So the swash zone is the area of the beach where the waves break on shore. And this turns out to be a very turbulent and hazardous place for animals to live. One of the big challenges for animals in the swash zone is that they have to move as the tide comes in and goes out every day, all day. So that's a big challenge to stay in the swash zone. And then there are a number of others as well. One is damage from mechanical action by the waves. So animals can become dislodged, they can get damaged, they can be swept out into the open ocean. If they don't move with the tide successfully, they can be stuck on dry sand and end up desiccating. So there are a number of different kinds of physical challenges. And then there's also a host of predators that are waiting to get them on the shore, predators that are waiting to get them if they end up in the open ocean. For example, with the sand crabs that I'll be talking about, there are some surf perch for which 80% of their diet is Pacific sand crabs. So these are some of the perils of the swash zone. As a consequence, the swash zone in general tends to be a pretty low diversity habitat. We just don't see all that many kinds of species in this environment. There are some, so here are some examples of the swash zone inhabitants that you may see when you go to the beach. So there are some there, and all of these species share one trait in common that enables them to persist and exist and succeed in this environment. And that is that they all burrow. So here are some of the signals of burrowing by some of these animals that you may see on the beach. So burrowing turns out to be key for survival, to escape predation and to escape the mechanical damage caused by wave action. Now, the superstar of the swash zone all along the Pacific coast is the Pacific sand crab, Emerita analoga. So this distribution map on the right here shows the range. So these lined areas are the range. These crabs occur all the way from Alaska down to Baja California. There's a break in their distribution, and then they occur again in South America. So they are a very ubiquitous and broadly distributed species. They exhibit sexual size dimorphism. So females are bigger than males. Females are typically anywhere from 20 to 30 millimeters in length, somewhere around there. And males are about 12 to 15 millimeters in length. So there's a big difference in size between individuals. They're also kind of funny looking to me. I had a hard time at first telling the head end from the tail end of these crabs. So this is the head end in this image below of a crab. You can see they have these long eye stalks. So their eyes are on these kind of like periscopes on these stalks. They have a couple of different sets of antennae, one for oxygen exchange and another for feeding, as we'll see coming up. So the head end of this crab often is mistaken for the tail end because they're unusual. They're also unusual in that they move backwards. They don't move side to side like most of the crabs we can think of. They swim, crawl, and burrow backwards. And when I say they are ubiquitous, one study estimated that there are more than 75,000 per meter of shoreline on the Pacific coast, including in the Central California coast area. So these crabs mate in late spring to early summer. They mate on the sand. So a female who has eggs yolked up will attract a bunch of males. They all kind of burrow into the sand. And then the males release a ribbon of sperm when they're mating. And the females hang on to the eggs. So these are a species that are known as a brooder. So they don't spawn, they don't release their eggs, and they don't gestate them inside. Rather, they carry them outside of their bodies. So if you grab a female Pacific sand crab and gently open up the tail or telson, you'll see these bright orange eggs if she has any eggs. And these eggs eventually mature and then hatch, and the larvae are released into the open ocean. If you ever collect a female that doesn't have eggs, the first clue that it's a female will be her size. If it's a crab of any size, it's most likely a female. But if you do open that telson or tail and you see these thread-like structures, which are called pleopods, that's another way to tell females from males. Those pleopods hold the eggs onto the female's body when she does have eggs. So she releases these larvae into the open ocean. Here are some drawings of what the different stages of the larvae look like. And they are pelagic. They go out into the open ocean. They can be distributed for hundreds and hundreds of miles. So there's a lot of mixing of populations all up and down the Pacific coast as a consequence of their larvae being out in the open ocean. And then they develop through these different stages and ultimately end up at the final stage, which settles on the beach, and then they stay there. They remain there throughout their lives. A female sand crab can live for 1 or 2 years in this area. The ones in South America have been shown to sometimes live as long as 3 or even 4 years. So these are filter feeding animals. I said that they have antennae that they use to feed. So when they're burrowed into the sand, they stick their eyes out so that they can see. They stick the antennae that are for oxygen exchange. They stick those antennae up and exchange oxygen. And then they use those large feathery antennae to filter plankton from the seawater. And then you can see periodically that this female is inserting that antenna into her mouth and eating the plankton that she's picked up on these feathery antennae. So as a consequence of being filter feeders on plankton, these animals end up accumulating whatever's in the water because they're constantly in the water and constantly foraging in the water. As a consequence, they are used as an indicator species for a number of studies of pollutants and toxins and things that we are concerned about. One of those is algal blooms. So sometimes we have red tides like the one pictured here. This is an explosion of plankton like dinoflagellates and diatoms. And some of these end up having this red coloration, which is why we call it a red tide. I think we had one fairly recently in this area. So if you heard about those bioluminescent blue waves that we had recently in oceans here, and then followed by the red tide, that is one of those algal blooms that I'm talking about. So one of the detrimental things that happens when we have that kind of a red tide is that those phytoplankton produce a phycotoxin called domoic acid. And it turns out that Pacific sand crabs are an abundant, good indicator species for domoic acid in the water. And domoic acid can bioaccumulate in animals that eat the sand crabs. It's more concentrated than it is in the sand crabs. And then in animals that eat those animals, it can become even further concentrated. So these kinds of red tides have been implicated in massive mortality in shorebirds and in sea otters and in shellfish. So if we're concerned about ingesting domoic acid, we could use Pacific sand crabs as an indicator of what the levels are like in the ocean locally at that time. So here's a study that was done on that, Emerita analoga, a possible new indicator species for this phycotoxin domoic acid. And this was right in California. Another way in which people are using Pacific sand crabs to study a really important pollutant is that they sequester microplastics. So microplastics are, as the name indicates, microscopic bits of plastic that result from the breakdown of plastic waste in our oceans. And these microplastics end up in all kinds of animals, including us and including these sand crabs. And researchers are looking at the effects of having these microplastics on mortality rates and reproduction in sand crabs. So these are not the topics of my talk today, but I just wanted to point these out as an amazing and excellent indicator species for all kinds of different studies. The Pacific sand crab is a very important part of an ecological food web that occurs locally on our beaches. So the sand crabs, as I mentioned, ingest the plankton that's in the water, and they themselves are an incredibly important prey species for a number of shorebirds and also fish like some surf perch. They're also ingested by some scavengers like this big crab, Blepharopoda, and I'll say more about that in a minute. Now, we're also part of this ecological food web. There are some hilarious YouTube videos of people trying to cook and eat Pacific sand crabs directly. It doesn't usually seem to go very well, but we do use them for bait much more successfully. And so annually, millions of Pacific sand crabs are used as bait to catch things like perch. Now, I said I'd talk a little bit about Blepharopoda, just in case you should encounter one of these. When my students did field work on our local beaches, they were working with these sand crabs. Sometimes they'd be holding a really big Pacific sand crab, like the one you see here in this image below. And then all of a sudden, one of these would walk by, and they're just like, what is that? It's gigantic. It looks like a spiny tank walking on the beach, somewhat menacing looking. It's a related crab that is a scavenger. They're out in the evenings, where they scavenge all the dead sand crabs that you've probably seen that accumulate on the beach. So I just wanted to point out Blepharopoda because I think it's a really fun animal to be able to spot on our local beaches. But I want to focus on the Pacific sand crab, Emerita analoga, and perhaps the behavior that we all know the most about these animals is that they burrow. And they don't just burrow, they burrow extremely quickly, and they burrow repeatedly throughout the course of the day as they're dislodged by wave action. As the tide changes, they constantly have to reburrow as quickly as possible. And burrowing is key, again, for survival in this species, because there are so many predators that are ready to catch an animal that's out of the sand, and even some that are burrowed in the sand. So our lab studied burrowing behavior and some factors that may influence burrowing time in Pacific sand crabs. So as I show you this image, I want to point out a few things. The first thing is that these folks are not doing it right. Pacific sand crabs burrow rear end first. So when you see a sand crab burrowing, it's the head that is sticking out and the rear end that's going into the sand first. So this is the appropriate way to burrow, should you want to try it yourself. The other thing I want to point out, of course, is that faster burrowing should be favored by natural selection. Selection should favor this adaptation, this trait of burrowing as quickly as possible, repeatedly, so that they avoid predation and avoid being sent out into the open ocean or ending up outside of the swash zone. The other thing I want to highlight with these images is that the sediment type that animals burrow in may have a significant effect on burrowing time. Imagine these crabs burrowing in really coarse sand that's very uneven in terms of grain size, like the one in this image, versus trying to burrow in very fine sand where the sand grains are really evenly sized. So our lab wanted to look at whether sediment type may affect burrowing time. The other major factor that we looked at in this study was whether parasites affect burrowing time. And we focused on one of the parasites that infects these sand crabs. They are hosts to a number of parasites, but probably the one that is best studied is this one. This is a thorny-headed worm. It's in its own phylum called Acanthocephala. So this is an Acanthocephalan worm. This is what the adult worm looks like. It has these spines all over it, which is where they get their common name from. But it's this life history stage on the right here, the juvenile stage. This is called a cysticanth. This is the life history stage that ends up in Pacific sand crabs. And so we wanted to see whether these parasites may affect burrowing time. And I'll talk in a minute about why we think this may be the case, that they would affect the burrowing time. So the parasite that we focused on, its species name is Profilicollisaltmani. And here's the complex life cycle of this parasite. It's a complicated parasite in that it requires two hosts in order to complete its life cycle. So the eggs of these parasites are found in the open ocean, and they are deposited into the ocean by birds pooping these eggs out. So these are fertilized eggs that are ready to develop into adult parasites. And when Pacific sand crabs filter feed, they end up inadvertently ingesting these eggs sometimes. And when they do, the eggs then develop into the juvenile cysticanth stage in the body cavity of the Pacific sand crabs. So the crabs are intermediate hosts. But in order for this parasite to complete its life cycle, to develop into adults and mate and then produce eggs, it has to make it into its final or definitive host. And that final host is a shorebird, typically some sort of a gull species or surf scoters. Those are the birds that it's been described in so far in the literature. So these cysticanth juveniles end up inside birds when the birds eat an infected sand crab. The bird eats the sand crab. The juvenile parasites end up in the intestines of the bird. They develop into adults in the intestines of the bird. They become these thorny headed worms, and they use those spines to latch onto the intestinal wall of the birds that they infect. There the parasites mate, the eggs are fertilized, and then they end up pooping out, the birds end up pooping out the fertilized eggs into the water, and the whole life cycle can begin again. I want to point out that other animals eat these sand crabs as well. And so unfortunately, sometimes these infected sand crabs are eaten by sea otters. And sea otters are referred to as a dead-end host. It's a bad situation for everybody involved. Of course, the crab dies because the sea otter has eaten it. The parasites cannot complete their life cycle successfully in the intestines of sea otters. So the parasite ends up dying as well without completing its life cycle to adulthood. And in many cases, the sea otter dies as well. And the otter dies from something called Acanthocephalan peritonitis. And I'll talk more about that a little bit. So here is an image that one of my skillfully taken image by one of my students through a dissecting microscope of a partially dissected sand crab. And you can see all of the parasites. So these white little rice grain-like things, they're all cysticanths of parasites. We found as many as 40 or 41 cysticanths in a single Pacific sand crab. So sometimes these crabs can have a lot of parasites. And it's not just sea otters that can suffer mortality as a consequence of infection. Sometimes even the definitive host birds, if they eat enough crabs, and if those crabs have enough parasites, the birds can actually die from this as well. And here's an example. This is from our vertebrate collection at Cal Poly in the Biological Sciences Department. And this animal, which you see mounted below, is a female surf scoter. And this on top here is her intestine dissected out. All these things you see, these things sticking out of her intestine, those are all Acanthocephalan worms, probably Profilicollisaltmani. She had hundreds of worms in her intestines and probably died from this infection. And in fact, these parasites have been implicated in episodic deaths of large numbers of surf scoters. So it's not always the case that this is a benign parasite that the animals can just deal with. Sometimes the infection can be fatal. A little bit about sea otters, because we all love sea otters, even though that also was not the focus of our study. So as I mentioned, Acanthocephalan peritonitis can kill sea otters as well. And there have been a few studies done on this. It's my understanding that it's young sea otters that usually end up being forced by competition to forage on the sandy bottom as opposed to foraging on sea urchins and things like that, which are preferred. So the young animals or other animals that are unable to compete for the best prey, they end up eating sand crabs. And if those sand crabs have these parasites, then they can get them as well. So here are some images of the intestines of an infected sea otter. So on the left here, you see an image of the intestine, and these arrowheads are pointing to Profilicollisaltmani parasites, of which there are tons in the intestines of this animal. And then on the right, you see the omentum, or the tissue surrounding the stomach and intestines of the animal. And you can see that there are a couple of these parasites that have broken through the intestinal wall. So that's the perforation that ends up being fatal. And it's that can cause somewhere between 16 and 20% of sea otter deaths in California are blamed on infection with this parasite. But going back to the life cycle of the parasite that's typical and that is successful for the parasite, I want to point out why it is that we thought the parasite infection may influence burrowing time. It would be an adaptation for these parasites if the crabs burrow slower, right? Because if they burrow slower, then they are more likely to be ingested by the shorebirds that the parasite needs the crabs to be ingested by in order to complete its life cycle. So it would be an adaptation for the parasite potentially to cause the sand crabs to burrow more slowly, and that way they'd be more likely to be ingested by a bird. It may also just be a pathological side effect of being infected for these crabs if they burrow slowly. But in either case, the parasite would benefit if infected sand crabs burrow more slowly. So with all of this background in mind, we address the following questions. First of all, we wanted to compare local beaches because they differ in their sediment types. And we asked, does parasite abundance differ among sites? And parasite abundance refers to the number of parasites per animal. Sometimes it's called parasite intensity. It's the same thing. We also asked whether parasite abundance differs with reproductive condition of females. Do females with eggs have different numbers of these cystacanthes in them than females without eggs? And finally, the main focus of our study, how is burrowing time of female Pacific sand crabs influenced by sediment, parasite infection, the site that they're in, and their reproductive condition? We focused on females simply because females were far more abundant when we performed the study than were males. And I want to point out that when I say we, I really mean they, because this work was done by excellent undergraduate students. These were the very first undergraduate students that I worked with when I started my job at Cal Poly, and they are amazing. They ended up in wonderful careers. Some of them are physicians, some are dentists, some are teachers, but they all did an excellent job on this work to the extent that they are co-authors on a publication to come from this work. They worked really hard. Sounded glamorous at first to be working at the beach and getting research credit for that. And then they realized how much really hard work is involved in collecting these animals and working with them. And so they did a fantastic job. So here are the field sites at which we did our work. They were in Avila Beach, Pismo Beach, and Hazards in Montana de Oro, all with very differing sand types and harshnesses of the environment. And we tested animals from all three of these sites in all three sand types. So the first thing we did was to quantify the characteristics of the sand type at each of these sites. So apologies for this small table, but I just want to highlight that we used a series of sieves and a standard methodology to determine the average sand grain size and also the variation in sand grain size of sand from all of these locations. So we took sand from Pismo, Avila, and then a couple of places in Montana de Oro, Hazard Canyon Reef, and also Spooner's Cove. And what we found is that the sand grain size increased going from Pismo to Spooner's Cove. So the sand is the most fine at Pismo Beach and the coarsest in Montana de Oro, especially at Spooner's Cove. And the variation in sand grain size also increased. So Pismo has very evenly sized sand grains, and in Montana de Oro, these sand grains are much less even. So there's a bigger range of sand grain sizes. And so we categorize the sand then as extra fine, fine, coarse, and extra coarse. Here are some examples. So here is a sand crab resting on Pismo sand, and here's a sand crab burrowing in a container of Pismo sand at one extreme. And then here is a sand crab resting in Montana de Oro and also burrowing in that extremely coarse sand. Having characterized the sand types, we then tested the burrowing behavior of female sand crabs from all these locations in all of these sand types. So the first part of our methods then was to time the burrowing of these sand crabs. We did this for each animal 4 times, once in each of the 4 sand types, in a randomized order, so that the animals didn't all see the sand types in the same order. So we randomized the order. We ensured the temperature was consistent during these trials, and all kinds of other controls were put into place. And after measuring the burrowing time, the animals were all humanely sacrificed. And in the lab, we measured their their body size, their standard length, carapace length. And then we dissected all of these crabs to count the number of cysticanth parasites in each crab. And we did this in the same time period at all of the different locations so that we minimize the chances of any kind of time pattern that we would have inadvertently generated otherwise. So here are some of the results we found. We found that these animals burrowed the most slowly in the coarsest sand. So they did all right in the extra fine, even the coarse sand, they were okay, but in the extra coarse sand, their burrowing was significantly slowed down. And I should point out here, we had 168 females total in our sample. We also did a side project. A couple of the students did a side project at the Pier in Avila. If you haven't had a chance to visit the Cal Poly Pier, I highly recommend it. There are periodically open houses, at least once we get back to normal activity, there will be open house events at the Pier. It's a great place to work and a great place to visit. And we got to do some work there. These students rigged up something that was really cool. They tethered individual sand crabs by just putting a little bit of glue on the carapace, and then they connected the tethered sand crab to a force transducer that translates their force that they're putting on the tether into a distance measurement so that we could see how deeply the crabs are burrowing. And they did this for crabs of different sizes, with and without parasites, with and without eggs. They did a large study. I'm just going to talk very quickly about a little bit of it. So they measured force as an indicator of depth of burrowing. And what they found is that the females burrowed more slowly and more shallowly in the coarse sand than in the fine sand. And this can be really important for the evolutionary fitness of these animals, because animals that burrow too shallow have been shown to be eaten by predators more easily than animals that burrowed more deep. So being in coarse sand is very detrimental for these animals. They can't burrow as quickly to get away from predators, and they can't go as deep in a given amount of time. We also found that infection with the P. altmani parasites slowed their burrowing as well. So what you're looking at here is parasite abundance per crab. So going from 0, these are animals in this first bar that didn't have any parasites. Some had 1 to 2, some had as many as 41, as I mentioned earlier. So even having a couple of parasites, even 1 or 2 parasites, slowed down burrowing. Certainly having more than that slowed burrowing further. So this is, you're looking at burrowing time here. So the larger the number, the slower the burrowing was. And this was regardless of sand type. So this is holding sand type constant. They burrowed more slowly. This is true of all the sand types. In fact, they burrowed more slowly the more parasites they had. We also found some interesting patterns with respect to females with eggs versus females without eggs. So non-ovigerous means females without eggs, and ovigerous refers to females with eggs. What you can see here from this pattern is that across all 3 sites, females with eggs had more parasites than females without eggs. That was an interesting pattern. What you probably also notice immediately is that there were way more parasites per crab in Montana de Oro. This is actually true for females without eggs and females with eggs. But what I want to point out is that the difference was also enormous at hazards in Montana de Oro. So the difference in parasite abundance for females with eggs versus females without eggs was also much greater in Montana de Oro. So harboring eggs and being in Montana de Oro, very, very difficult ecological environment for these crabs. So females with eggs had more parasites. I'm not showing data here for this, but interestingly, females with eggs burrowed faster than females without eggs when holding parasite abundance constant. So regardless of sand type and regardless of parasite abundance, females with eggs burrowed faster than females without eggs. And I'll talk about what these results may mean coming up. So to summarize the conclusions from this study, first, the more parasites the crabs harbored, the slower they burrowed. And as I mentioned earlier, this may represent a really cool adaptation on the part of parasites. Maybe they do something to the nervous system of these crabs that cause the crabs to burrow more slowly and manipulate these crabs so that they're eaten more easily by shorebirds. It could be that, or it could simply be a pathological side effect of having these parasites, having this infection that causes the crabs to slow down. Either way, this is helping the parasites complete their life cycle. And crabs burrowed slowest in the coarsest sand, regardless of how many parasites they had. So independent of their parasite abundance, the coarsest sand was the hardest for them in terms of burrowing. I want to show you a brief video clip of how these sand crabs burrow from a mechanistic standpoint. This is not our work, it's the work of someone named Ben McEnroe at UC Berkeley, but let me just play this video clip for you.
So the crabs have become tiny engineers. They stir up the wet sand with their tails, making it easier to move. A process called liquefaction. Then they push the loose sand up toward the surface by handing it off between their 5 pairs of legs. See? Kind of like a conveyor belt. The waves never rest. So the mole crabs do this day and night as they're tossed and tumbled. But they take it all in stride.
All right, so this liquefaction of the sand that has to be accomplished by these sand crabs, that's probably what's so tough in the coarse sand compared to fine sand. If any of you is interested in this work, here's an abstract of the work that Ben McEnroe and his team is doing on this. So they're approaching this from kind of an engineering standpoint as the mechanics of burrowing. And interestingly, it looks like this work may have some relevance for designing robots with legs. So you never know where this kind of science, basic science, will take you in terms of applications to other areas. So I will also want to point out that we found that the beaches that we studied vary in terms of the harshness of the ecological environment. Montana de Oro has the coarsest sand, and it also has the highest parasite abundance, especially for females with eggs, compared to Avila and Pismo. So this environment is really different for the sand crabs than Avila and Pismo are. And it's possible that the high parasite abundance is driven by slower burrowing, right? Because the sand is coarse, the crabs can't burrow as quickly, and maybe that is what contributes to the parasite abundance being so high, because birds can eat them more easily there, and the parasite can therefore complete its life cycle more easily in Montana de Oro. However, there are other possible explanations as well. It could be that differences in bird abundance are driving the differences in parasite abundance. This is a really cool study done in 2008. This research team looked at the effects of El Nino on this parasite life cycle. And what they found, one of the things they found is that, so if you compare the non-El Nino life cycle on the left to the El Nino life cycle on the right, what they found is that there were fewer parasites per sand crab during El Nino years. And they hypothesized that this could be driven by bird abundance. So the El Nino storms cause habitat destruction that leads to poor nesting success for the birds, that can lead to fewer birds in the El Nino periods of time. And fewer birds means fewer eggs in the water of the parasites, and that means lower parasite abundance in the sand crabs, in contrast with non-El Nino years when you see that they have more parasites. So it could be that there's something going on. I don't know readily the differences between Montana de Oro and the other sites in terms of bird abundances, but that could also be helping to drive the pattern that we see. I want to point out that in general, beaches can be categorized on a continuum. So some beaches are said to be more dissipative beaches, characterized by having this long swash zone or large swash zone. I think Pismo can be characterized as a dissipative beach. So the action of the waves is dissipated, and that leads to finer sand grains and maybe a less harsh environment for these sand crabs. In contrast, a place like Hazards in Montana de Oro may be more of a reflective beach, which has a very small swash zone, strong wave action, very coarse sand, and that may be why the environment is harsher there. We also found that females with eggs were more likely to be parasitized, and this was across sites, even though it was the most pronounced at Montana de Oro, and they also had more parasites once they were parasitized. This suggests a few possible explanations. It's possible that there's a trade-off with the immune system. So females who are yolking up, who have yolked up eggs, may be putting so much energy and resources into those eggs that they are not able to maintain their immune systems at the same level as females without eggs. And that weakened immune system may allow the eggs of the parasites that they ingest to become cystocants instead of being encapsulated and destroyed, right? So that it could be that kind of a trade-off that females are making. It also could be that females who are yolking up eggs simply just eat more. And the more that these animals eat, the more likely they are to ingest parasite eggs. It also could be the case that females with eggs are older, even though we may be talking about 1 year old versus 1 year and a few months old. That may make the difference in terms of accumulating more parasite eggs and more cystocants in their system. Interestingly, females with eggs burrowed faster than females without eggs, which is not necessarily what we would expect. We may have expected that the eggs would slow them down or that the energy trade-off would slow them down, but it's possible that natural selection favors females when they have eggs burrowing as quickly as possible to protect those eggs. So they may be trading off their immune system in favor of burrowing more quickly in order to protect their offspring. I do hope that someone else takes up this study. I think it would be a really interesting future study to do. Our lab doesn't have any plans to work with this species, but I hope someone does. And I hope they look at why the crab's eggs are so orange. So my collaborator on this study, Dr. Larissa Redivo, she and her graduate students have looked at the eggs of the sand crabs and shown that they have pigments called carotenoid pigments. And carotenoids are really important in the diets of all kinds of animals. They are responsible for the yellow, orange, and red colors of lots of different birds and fish and invertebrates that are really familiar to us. They also are the pigments responsible for why carrots are orange and tomatoes are red, and all kinds of other fruits and vegetables, why they get their colors. So carotenoid pigments are an important part of the diets of all kinds of animals. We as animals cannot make them ourselves. We have to get them from our diet. And one of the reasons that we're advised to eat a rainbow of different kinds of produce is that we then will acquire lots of different carotenoid pigments. They are also what makes egg yolk yellow. So you see here a bunch of eggs with differing amounts of carotenoids in the yolk, and you can see that the color is most intense when there are the most carotenoids in those eggs. The reason we're all advised to eat so many carotenoid pigments and why they're so important is that they are really important for our immune system. So although their availability is limited for animals across the board, they're important because they scavenge free radicals and they prevent damage to our DNA. So that's why we're advised to eat them. That's why animals ingest carotenoids. And they also put carotenoids into their eggs in order to protect their offspring from this kind of damage. And then for animals that live in places where UV damage is a concern, carotenoid pigments can also limit UV damage to the eggs. So female sand crabs are putting these carotenoid pigments into their eggs, making them so bright orange in order to protect them, in order to help their chances of survival and hatching into larvae. But I wonder if they're suffering a different kind of trade-off as a consequence of this. Because when we look at people who use sand crab lures, one of the things we see in common is that these lures all have that bright orange color. So people know that that bright orange is something that predators like fish cue in on. And so I wonder if female sand crabs who have particularly bright eggs are facing a trade-off. Their eggs are going to be particularly protected, but they may also be extremely vulnerable to predation. And oftentimes when animals eat the eggs, they actually are aiming for the eggs themselves. They're cueing in on that orange color, I think. They also end up damaging the crab to the point where it can no longer burrow successfully because that tail end of the crab is damaged, and that ends up killing the crab as a consequence. So I wonder if animals like surf perch are also cueing in on this orange color of the eggs. I think it would be really interesting for someone to study whether there's higher predation on females with eggs, and especially females with more orange eggs, such that they face a trade-off. So with that, I would like to acknowledge the Cal Poly Center for Coastal Marine Sciences and Cal Poly for financial support. My collaborator on this study, Dr. Larissa Redivo, who's a professor in the Biological Sciences Department as well. Jason Felton and Tom Moylan at the Cal Poly Pier. Tom runs the Cal Poly Pier. And then a host of wonderful former undergraduate students who are the ones who really performed this study. And thank you so much for attending this talk. I really appreciate it. And here's my email address in case you want to get more information as well.